Hello everyone, my name's Keith Hunter. I'm Professor of Head and Neck Pathology in the University of Liverpool. I'm a clinical academic histopathologist with an interest in cancer biology, and the lecture today that we're going to cover is the cancer and host cell interactions, particularly focusing upon the role of the tumour microenvironment. During this lecture I'm going to revise the hallmarks of cancer. These are basic features of cancer, particularly the cancer cells themselves, but we'll see that this has a much wider application into other elements of the tumour. And it's very important to understand the tumour microenvironment, which is the overall context in which cancer develops in relation to this. So then I will introduce the concept of the tumour microenvironment and explain what that means before we go on to have a look at the components of it and how the interactions between these components affect the tumour and how cancer develops and progresses. Then towards the end of the lecture, we will have a look at some of the clinical implications and some of the new and interesting areas in which we might be able to therapeutically interact with the tumour microenvironment. A few things that we'd hope you'll be able to do by the end of this lecture, including demonstrate some understanding of the concepts which I have covered. I'm sure very many of you will be aware of the whole concept of the hallmarks of cancer. This was a process which was outlined initially by Hanahan and Weinberg about 20 years or so, but this has been built upon in a number of ways, adding further capabilities of cancer cells and from their microenvironment in terms of the features that cells develop as cancers progress. And there are a whole number of these, as you can see here. We're now at a point where there are about 14 or so of them from the initial 6. And whilst a lot of these capabilities refer to the cancer cells themselves, there are a number of these, as we will see as we go through this lecture, are very much, if not solely related to the tumour microenvironment, for example, like inducing or accessing vasculature or tumour promoting inflammation. The tumour microenvironment plays an important role in influencing the cancer cells themselves through these various mechanisms. And so the overall concept of the tumour microenvironment is that a cancer is not just the malignant cells themselves. There are a whole number of other cells which are involved and are really very important in the development of malignant neoplasia. Some of these components are cellular. Some of them may well have been resident in the area where the tumour has developed. We'll see, for example, some of the fibroblasts have already been there. But others are infiltrating cells, and that not only includes the cells of the inflammatory component and the immune component, but even perhaps some of the fibroblasts and the endothelial cells, which are going to bring a blood supply into the area. They come into the tumour to populate it and to increase the richness of the cellular component. In addition to the cells, there is extracellular matrix, and we're going to explain a little bit what that means, but it's the background chemical, structural proteins, glycoproteins and so on that make up the background in which these cells survive. And then a lot of the cells, be it the tumour cells themselves or the cells of the tumour microenvironment, will secrete a number of factors, chemokines, cytokines, growth factors and so on, and they are the prominent part of communication within the tumour microenvironment. And so overall, that gives a very rich cellular and extracellular environment, a context in which the cancers develop. And it's not just a bystander, it just doesn't just develop as the tumour cells get going. It is a key important part, an active promoter of cancer progression. And the cancer cells themselves and these various components within the tumour microenvironment reciprocally interact with each other, and so that they will have feedback loops and so on that will increase the intensity of that communication. And you can see on the picture on the right here some sort of idea of the number of different types of cells. It is not just the tumour cells. The tumour cells, these are these grey cells in this, but there's a richness of blood vessels and fibroblasts and other immune cells which are making up the whole of the microenvironment. And that develops at the primary site when it starts perhaps even as a pre-invasive component through to the development of the invasive tumour microenvironment, and then to some extent that is recapitulated when a tumour metastasizes, perhaps be that to a lymph node or to another body site. The microenvironment, the context in which the tumour cells are and survive and grow and progress, is very important. So here's an example of histology. This is my own particular area where my clinical work is in histopathology. And this is an example from a head and neck cancer. It's a squamous cell carcinoma of the oral cavity from the tongue. And in the center, you can see that there are cells which are present, which have the epithelial cells. They are keratinizing in the center here. So these are the malignant cells with their nuclear and cellular pleomorphism and so on, the features they expect to see of malignant cells. But there's a whole load of tissue in the background of this, and this makes up the tumor microenvironment. There are the immune cells, and there are varied ones. There are perhaps some lymphocytes and other cells, and they're macrophages. There are some spindle-shaped cells and even some stellate-shaped cells in areas, and they are probably fibroblasts, and we'll talk about them. There are blood vessels. A new tumour requires a blood supply, otherwise it becomes very quickly hypoxic, and there are cells which constitute these blood vessels. And in the background, on this H&E stain slide, are these areas of fibrillar material. It's making up collagen and various other proteins and glycoproteins in the background here that make up the extracellular matrix. So it's overall a complex mixture of these cellular and acellular components. So how important is this? Well, I indicated this in an earlier slide that in fact it is vital to the development of malignant tumours. And from this diagram here, you can see that right from the very beginning of the development of a cancer, the stroma, the tumour microenvironment, if you like, and the stroma is sometimes used as a synonym for that because it is this background connective tissue which surrounds the tumour cells. Then it's there, it's important at the initial point, perhaps even in pre-malignant point, and then in the development of the primary tumour stroma as invasion begins. And there is that communication. You can see the arrows are running between the tumour parenchyma, that's the tumour cells themselves, and the tumour stroma or the microenvironment. And these communications run both ways and they mutually promote each other. The cancer cells will be promoting the tumour microenvironment, which when stimulated in turn will promote the cancer cells to become more aggressive, become more invasive, perhaps more motile, and so on. And so that is there present at the development of the primary tumour. But then when metastasis occurs and the tumour spreads to another part of the body and there it seeds and begins to grow again, it's not just the tumour cells, there has to be the induction similarly of a tumour microenvironment that allows the cells to grow and to develop, to survive and to progress. And that is the way when you get successful metastasis, that there has to be an associated tumour stroma or tumour microenvironment that allows that to occur. And so it is, as the tumour continually spreads, that is something which has to happen. Now, as we will see as we go through this, the tumour microenvironment is not just a complex connective tissue in which the cancer develops, as if it's like a normal connective tissue in the background. And you can see in this diagram here on the left that there is your normal epithelial cells with the basement membrane and then underlying connective tissue. Within this case, it's got mesenchymal cells, it's got fibroblasts and so on. But the extracellular matrix is there. The cells, they are fundamentally altered. They are activated cells. And perhaps not even just the normal physiological activation, because a lot of these cells become activated in wound healing. But it's an alteration in the extracellular matrix. It becomes rigid, it becomes stiff. The cells are activated in a different way so that they become more tumour promoting. And other cells also come into the tumour microenvironment which fortify that. And so it's not just a normal connective tissue. Yes, it does take some of the concepts that we know and we understand from wound healing, and some of these processes are used in the development of the tumour stroma, but it's not like wound healing where it's switched on and switched off again when the wound is healed. This is something which keeps on going. And so some people have termed cancer the wound that never heals, and this is part of the reason for that. So here, I've just introduced to you the idea of the tumour microenvironment, and just take a break now and just review the material that we've been through, the whole concept that there is a supporting stroma background connective tissue which contains cells, which also contains acellular or extracellular matrix, and that supports the development of a malignant tumour by communication both directions between the tumour cells and the various elements of the tumour microenvironment. And now, when we step on from this, we're going to have a look in detail at some of the components of the tumour microenvironment. So what about the cellular components? The cellular components of this, well, the immune cells are very important. And there are both innate immune cells, that is a part of the innate immune system, the non-specific immune system. So we're talking about neutrophils and macrophages and other dendritic cells. And then there are cells from the adaptive immune response. Now, those which are particularly specific, responding to a particular antigen, and these are particularly T cells and B cells, and to some extent natural killer cells, although they probably sit in the middle between the innate and the adaptive immune response. So there is a rich immune component to the cellular part of the tumour microenvironment. And then there are what we would perhaps classically think of as stromal cells. These include fibroblasts and endothelial cells being the cells which line blood vessels. And then there is a mixture of other cells, adipocytes, fat cells, nerves, and so on, but also non-differentiated mesenchymal cells as well. And these may then differentiate into various lineages that we see in the developing tumour microenvironment. And all of these cells, as you can see in the diagram here, they are there together in close association with the tumour cells. And the main communication that we have is sometimes by direct communication between the cells, by contact, but very often by the release of cytokines and chemokines in order to stimulate various signalling pathways and processes within the other cells in the tumour microenvironment. And this brings me to the next point, which is the fact that communication is key to making this whole thing work. This is just a very select examples of some of the chemokines, cytokines, and growth factors and so on, which are involved in the communication between the various component parts of the tumour microenvironment. You can see the inflammatory cells and all that they produce. But very often, another thing which you will notice here is that, as I've already said, the communication is reciprocal, it's bidirectional. Tumour cells will produce a mediator which will act on various elements of the tumour microenvironment, which will have an effect, but also will in turn stimulate further chemical mediators, which will then in turn feed back onto the cancer cells and alter their properties. And so the whole thing is a very rich matrix, if you like, of communication between all these different cell types. And in the centre of this is the extracellular matrix, and perhaps even the bone marrow, because some of the cells that are important and come in to the tumour microenvironment may well have originated in the bone marrow. So there's quite widespread signalling which may actually extend beyond the bounds of the tumour. So what about the individual cell types? Well, one of my main interests, from a research point of view, is the function of fibroblasts in the tumour microenvironment. I'm sure you'll have been introduced in time past to fibroblasts and their function, how important they are in the overall structure of connective tissue and the formation of the extracellular matrix. They produce collagens, they produce ground substance and so on, which gives the basic structure of connective tissue. And so formation is important, but it's not a static thing. It's always being turned over. So the extracellular matrix is being turned over by fibroblasts. Now, fibroblasts also become activated in wound healing. They become secretory as well as formative cells in terms of producing proteins. And there are various subtypes which are now being understood in more detail, which control various aspects of wound healing. And these are very important because they're very often also seen in the activation of fibroblasts in cancer. The fibroblasts that we find in cancer, we tend to term as cancer-associated fibroblasts, or sometimes referred to as CAFs. Where do they come from? Now, the diagram below shows that On the left, there is a normal fibroblast, and some do come from normal resident fibroblasts in the tissue. But there are a whole number of other areas, and they can come from the epithelial cells, that is the process of EMT, epithelial to mesenchymal transition. And some others are coming from mesenchymal stem cells from the bone marrow. You can see that at the bottom right. And other cell types can also undergo this transdifferentiation to become CAFs, cancer-associated fibroblasts. And they become activated. They've got lots of stress fibers, actin-related fibers in them, and you can see in the fluorescent image on the right there, activated fibroblasts. These are in cell culture from a tumour. So what do these cancer-associated fibroblasts do? Well, they do various things. The diagram here shows a number of the features that they have which are involved in tumour progression. Some are tumour suppressive, and there may be various elements of the function of fibroblasts which do suppress tumours. But once they are properly activated, in general, the effect of CAFs is tumour promoting. And there are variable activation states. Some of them become particularly contractile. We sometimes refer to these as myofibroblasts because they've taken on muscle-like appearance, and they express alpha smooth muscle actin. Some of these, and also some other subtypes, will also be producing a lot of cytokines and chemokines and growth factors. And you can see from the diagram that there are a lot of growth factors. For example, fibroblast growth factor, FGF, is there a number of times, vascular endothelial growth factor, and also chemokines like IL-6, IL-10, IL-1 beta, and so on. And they contribute to a number of the cancer hallmarks, because you can see that back, and we'll talk about that in a minute, about how they contribute to the progression of cancer. But we're now beginning to understand that there are perhaps even more subtypes, and there are some aggressive subtypes of fibroblasts, some of which may well particularly been associated with epithelial to mesenchymal transition. They're also very much associated with a markedly altered extracellular matrix, which are very much contribute to more rapid progression of the cancer. Because they secrete a large number of chemokines, they're also very responsive for the recruitment and activation of inflammatory and immune cells within the tumour microenvironment as well. And as we will see later on in the lecture, they may well also be responsible through a number of different mechanisms for modulation of responses to cancer therapy. So how are they contributing to cancer? And you can see this is a very complex diagram, but you can see here that there are a number of different areas looking at particularly the communication between CAFs and the other cells in the tumour microenvironment. So for example, there are some areas in which CAFs are going to inhibit. You can see here that these are the red lines with the bar at the end. CAFs will inhibit cytotoxic T cells. We will see that is very important in reducing anti-tumour immunity. They will affect other factors like macrophage polarization. Again, that's not helpful in terms of mounting an effective immune response to tumours. They will promote monocyte function. They will produce a lot of chemokines which will act on macrophages, other white blood cells. They are going to promote angiogenesis because they will promote the influx of endothelial cells and also the recruitment of other immune cells. So it's a very complex diagram, but you can see just exactly, and you can pick up when you look at the diagram on the right, just how much these individual communications between cells are contributing to the hallmarks of cancer. And some examples are marked up on here. This is an older version of the hallmarks of cancer when it was actually just reduced to about 10 features rather than the 14 we have now. But you can see how a lot of the chemical mediators which are here are very much involved in the promotion of cancer via these cancer hallmark features. So, well, that's all very complicated. What about looking at it on a very simple basis? Is there anything we can look at in tumours which tells us that this is actually really something which is important? And there is. And this is a very interesting paper which was published about 10 years ago. And it was looking at a number of oral cancers, oral squamous cell carcinoma. And If you take a marker of fibroblast activation, I've mentioned alpha smooth muscle actin, if you take that as a single marker and stay in a whole load of oral cancers, what you get is a predictor which is actually more predictive of death than any other clinical factor that you might wish to bring in to the analysis. So it's more important than cancer stage or anything like that. And if you have a high alpha SMA expression, so lots of activated fibroblasts, your mortality is going to be much higher than if you had a very low alpha smooth muscle actin expression. So that tells us that fibroblasts within the tumour microenvironment are doing something very important in determining the progression and the outcome from cancer. Now, related to that is the function of the extracellular matrix. This is a complex chemical mixture of structural proteins like collagens, glycoproteins, proteoglycans, chondroitin sulfate, and so on, and polysaccharides. It's a dynamic thing. It is undergoing constant remodeling, and it has a number of important features, as you can imagine, in wound healing and in the maturation of wounds and of scars, but many of these also come over into the development of the tumour extracellular matrix. And so there are important things. It is involved in communication. There may be direct contact between cells and various proteins or proteoglycans, and that will trigger signaling pathways. So that can be either direct or indirect. A very interesting function of the extracellular matrix, as you can see in part B in the diagram there, is that a lot of growth factors, although they are secreted from cells, they get trapped in the extracellular matrix and are not allowed then to act on cells. But if the tumour microenvironment, the extracellular matrix, is being turned over quickly, it will release growth factors which can then act on cells, and that pushes the cancer to progress. Another very important part is the physical barrier, and that's related to the stiffness of the tissue. If you have a thick layer of extracellular matrix and lots of collagen, well, then it's difficult for infiltrating immune cells to get in. And then another part of that is the fact that although there may be a physical barrier, if it's being turned over all the time, what you allow is then passages for invading cancer cells to grow and to progress and get through the tissues. And so this increase in turnover and remodeling happens in cancer, and it facilitates a number of different features, as you can see on the right in the diagram here, including facilitating of growth factor signaling, changes in the mechanical properties of the tissue, which again are very important in allowing tumour cells to be able to grow and progress through the tissue. So that's the fibroblasts and the extracellular matrix, which is largely a fibroblast product. We're going to move on and talk now about the immune microenvironment. So malignant tumours contain immune cells to varying amounts. In general, what we say in tumours is that the adaptive, that's the specific immune response, is inhibited. So normally you might expect that the immune system would see cancer cells, and we'll explain why in a minute that might be the case. But cancer cells develop ways of being able to locally shut down the immune system, which means that they are not seen by the immune system and therefore are not attacked and are not killed. The cells which are involved, you can see in the diagram on the left here, there are a number of them which are innate immune responses. And so that's those which are largely responsible for various elements of the inflammatory response. So we're talking about granulocytes and macrophages and so on, neutrophils. And then on the right is the adaptive immune response. Specific B cell function, producing antibodies which are going to bind to a specific antigen, and T cells also with the CD4 or CD8 cells, helper and effector cells, which again will bring specific effector functions, and in this context hopefully cell killing to the tumour cells. Again, how do we know this is important? And there are a number of different papers that could have shown this. This is one of our own, again, from a head and neck cancer patient. And we were looking at the patterns of expression in a whole range of these different tumours and looking for patterns which would predict survival. Those who did worst, as I've indicated, are those who had lots of smooth muscle actin. They had what we call the FA. They had a lot of activated fibroblast-related genes. But there are a lot of others which separated due to their innate immune response dominated features or adaptive immune response dominating features. Those who had an adaptive response dominated expression profile did better than those who had a non-specific and innate or inflammatory type response. And so we see here that having an active adaptive response to tumour from the lymphocytes within the tumour microenvironment is something which you want against a cancer, but it is something that cancer develops means to avoid. But innate immune cells are there, and macrophages and others such as neutrophils are very important. There has been a lot of interest over the last number of years about tumour macrophages. They are important cells. Macrophages are very important in wound healing, again, in terms of their phagocytosis and removing debris, and they also secrete a number of chemokines and growth factors which control other cells in the healing wound. And similarly in cancer, they're phagocytic, but they're also very secretory. And you can see in the diagram here that there are, again, a large number of growth factors and cytokines that are being produced by tumour-associated macrophages. That's what TAM means. And they are promoting various elements of the progression of a malignant tumour. Others, such as neutrophils, aid in extracellular matrix remodeling and signalling. They may well release factors which will promote epithelial cells to undergo this transition to a mesenchymal phenotype, and so on. So they're very important, but they're not really involved in specifically targeting the tumour cells and clearing them. That would be a function of adaptive immunity. And in the diagram on the left here, on the far left, you can see the innate immune response is not specific, whereas the adaptive immune response brings the whole concept of antigen specificity into the mix. As tumour cells grow and develop and progress, they become more genetically abnormal, and I'm sure you're aware of that. And part of the process in which they do that is that they change their antigenicity. They make what we call neoantigens, tumour neoantigens, and these can be processed and presented on the surface of antigen-presenting cells, with the idea that there would be a signal that this is not self, it's not normal, it needs to be targeted. And if that's the case, then the specific immune system, the adaptive immune response, should be able to identify tumour cells and target them for death. So again, in the tumour microenvironment, is this important? Does this actually relate to how tumours are clinically? And in the picture on the right-hand side, you will see it's another head and neck cancer specific example. But in this case, you will see in the cancer, some of which have human papillomavirus, some of which don't. But in general, you'll find that the tumours which are tumour-infiltrating lymphocyte, TIL, high, that means that they've got lots of tumour-infiltrating lymphocytes, do much better in their survival than those which don't. So if you take an HPV negative cancer of the head and neck, if you are HPV negative and TIL high, your 20-year survival is about 75%, but your 15-year survival, if you're TIL low, is probably about 38%. And so you can see it almost halves the survival. So these are doing something very important within the tumour microenvironment, which affects the progression of the cancer. But the dynamic interplay between all these cells in the tumour microenvironment is very important. There are elements of it which are going to be tumour suppressive, want to damp down the tumour. And these are those which are related to having specific T helper cell populations, cytotoxic CD8 cells, and so on, that are going to target tumour cells after identifying them as with foreign antigens and being able to kill them. Whereas there are elements within the tumour microenvironment which can be tumour progressing because they damp down the immune response and allow the tumours to progress. So for example, if you have large numbers of Treg cells or M2 macrophages, you will have what we call an unfavourable environment, which I shall show you in a minute. And so the evasion of the immune response can be due to a lack of immunogenicity, but in many cases it's due to inhibition of the immune response. The CD8 cells just never get in there, and that may be because of the fibrosis of the extracellular matrix. And there are other elements in terms of which effector cells can be inhibited and so on, which I'm sure will be covered in greater detail in terms of the function of the immune system in cancer. But overall, it gives an idea that you can have a favourable tumour microenvironment in terms of the immune microenvironment, one which will kill off cancer cells, or an unfavourable one which will promote cancer progression and make sure that the patient has a worse prognosis. And just very briefly, that brings us to immunotherapy. And there are various means of being able to increase the adaptive immune response in cancer, which are very important in how we might modulate the tumour microenvironment. Immune checkpoints, which are related to the function of CTLA4 and PD-L1, which will make sure that the cytotoxic T cells are inhibited. You can make drugs to inhibit that, which might activate the T cells again. And there are other ways such as transfer of T cells like CAR T therapy, cancer vaccines, and elements which might target other parts of the tumour microenvironment. So if it is the fibrosis of the extracellular matrix which is inhibiting access of cells, if you can target the fibroblasts, you might also be able to facilitate the function of the tumour immune system. What about angiogenesis? Well, angiogenesis is very important in the development of cancers. They need a blood supply. And so you can see from the diagram on the right here, as the tumour progresses, that the endothelial cells begin to divide, largely driven along by the lack of oxygen and the needs of the tissue, and they start to sprout. And so you then get the development of a tumour vasculature, which is not like a normal mature vasculature. They tend to be leaky and so on. They're not particularly good vessels. But it's very important in terms of making sure the tumour has the nutrients and access to growth factors and oxygen and so on to be able to survive. But a tumour angiogenesis and a microcirculation is also a route whereby when the tumour cells themselves are growing, you get access to the blood supply for that. So the facilitation of dissemination of cancer cells is an important function of angiogenesis. And pericytes as well, they may well be an important contributor to cancer-associated fibroblasts. And so the background function of endothelial cells and pericytes is also play an important part in the microenvironment. But as I indicated, this whole area of the cellular and extracellular components of a cancer is very important because nothing stays the same. This is a constantly changing and evolving environment. There is fluctuations in the oxygen tension, so you get hypoxia. And the diagram here shows, again, a number of tumour-promoting areas where you get hypoxia within the tumour, and the tumour microenvironment plays a large part in that, driven through signalling factors via like lactate and HIF1 alpha. You get fluctuations in pH, fluctuations in nutrients and nutrient supply, perhaps related to the blood vessels as well. And then once you put on top of that the effects of treatment, be that radiotherapy, the after effects of surgery or chemotherapy, you again change the tumour microenvironment, and the changes which are in that due to the treatment can also promote the development of cancer and promote resistance to further therapies. So that has really been through step by step the various components of the tumour microenvironment. And in the last section, I just want to talk about two main things, about the clinical effects and also what we might do about this therapeutically. So what about the clinical relevance overall of the tumour microenvironment? Well, there are two things. The first of these, which is no surprise given what I've talked about in the rest of the lecture, is that the tumour microenvironment is very important in the promotion of tumour progression. And you can see in the diagram here, as a tumour progresses from the very earliest invasive stage, and there are some fibroblasts around that, activated stellate cells and other cells. Once it progresses and it starts to grow, you factor in hypoxia, you begin to get the production of a number of cytokines. The extracellular matrix then changes, so you get an altered extracellular matrix, which may become stiffer. There are then the development of blood vessels, and the complexity of the tumour microenvironment grows. But with that comes the increased fitness of the cancer itself to grow and to divide and to progress at the local site, but also comes with it the accumulation of the abilities to be able to disseminate, be that by lymphatics or via the blood supply. And the tumour microenvironment and the various component cellular parts of that and the extracellular matrix play a large part in that, in allowing cancers to progress. The second main clinical effect that I wanted to bring before you was the whole concept of the tumour microenvironment and resistance to therapy. Now here you can see 5 different examples in the diagram, a really very interesting diagram which shows how the various component parts of the tumour microenvironment might actually make cancer therapy difficult. And in part A you see the extracellular matrix, the stiffness of the tissue acting as a barrier to the drugs itself, or perhaps to, as I indicated, to cells getting into the area. They might respond differently to chemotherapy and radiation. So you can see in part B that the drug or radiation is having a negative effect on the cancer cell, but it's having an effect on fibroblasts or other cells in the tumour microenvironment, which stimulates them to produce factors which will allow the cancer cells to be resistant to the drug and or radiation. Similarly with targeted therapy, if you target something which is affecting the cancer cells, but then the tumour microenvironment cells find a workaround, which means that they use a different chemokine to be able to stimulate the tumour cells. Again, you begin to get resistance even to targeted therapies. Similarly with hormone therapy and immunotherapy, the modulation of various factors within the tumour microenvironment, be that within fibroblast endothelial cells, inflammatory cells or immune cells, are going to contribute to the development of resistance under therapy. So that brings us to the last part, which is that, well then, surely in that case it is as important as targeting the tumour cells to find ways to target the tumour microenvironment. And I think it is important. And there are a number of different ongoing areas, some of which are areas of research in my own laboratory and others across the university, where we're looking at being able to target various component parts of this. Can we target activation of fibroblasts? And we're finding new small molecules which may be able to do that. Can we target the tumour vasculature to make it even more leaky and to contribute to even more hypoxia, which may result in the death of tumours? The lymphatics, anti-inflammatory inhibitors, and also the use of immune checkpoint inhibitors to reduce the inhibition within the immune component. And so there are lots of different examples of that. This is a very complex diagram, but if you just stop the progression, the slides going forward, and you'll be able to see and have a look at this, maybe have a look at it in the PDF, that it summarises a lot of current research which is going on in all the different component parts of the tumour microenvironment, where you can get all the different drugs which are being used. Some of them are very specific to particular pathways, whereas others are very generic. For example, over on the left-hand side, you'll see things like retinoic acid and so on. Not specific in terms of its effect on a particular cell or particular pathway, but can be very useful. And we're looking at reprogramming various elements of cell type and also the development of the tumour microenvironment, which can be very useful if used in addition to anti-cancer therapy for the tumour cells themselves, and together we can have a greater effect on the tumours. So what about overall summary? Well, in summary, the tumour microenvironment is very important in cancer development and progression. They have cellular component and an extracellular component, and there are multiple roles which have been described for each. And the important part is this is bidirectional reciprocal interactions between the tumour cells. We know that it has an effect on progression of cancer and cancer treatment efficacy, and we are trying, as we move forward in research in Liverpool as much as anywhere else, to look for new ways of targeting the tumour microenvironment. So that's all I wanted to bring to you in the lecture today. These are my contact details. If there's anything I've not made clear, please do contact me. And if any of you are interested from a research point of view in knowing what we do more, please do contact me.